Thank you, Neil, for the introduction, and thanks to the organizers for inviting me to present our newest data on the RNA in nanomedicines. We are a biotech company which is fastly growing, located in Mainz, Germany, not so far from here, also at the Rhine River. We are interested in several aspects of immunotherapy with different therapeutic and diagnostic approaches. Here I just would like to focus on, do we have a pointer? Oh, green button. Ah, yeah. Here I would like to focus only on mRNA-based nanomedicines, where we would like to interfere in, with an immunological route for cancer therapy. The foundation is that it is well known that tumors can be discriminated from healthy cells by their tumor-associated antigens, which can be also recognized by the immune system and attacked. Our aim is to stimulate and increase the immune response against the tumor-associated antigens by our medication. This we could do by a conventional vaccination, but we use RNA as a drug which then codes for tumor antigens, and the RNA needs to be delivered to antigen-presenting cells, dendritic cells, as they are resident in the spleen. And if you do this, you don't express only the antigens at the disease. You initiate a whole cascade of immune responses which all synergistically interact with each other to fight against the tumor. Now the task is how to deliver the RNA to the dendritic cell, as it's a very large, highly charged and labile molecule. There are two routes of doing this. One is by local injection into the lymph nodes, and one is by systemic administration by intravenous injection of lipid-based nanoparticles, which then selectively target antigen-presenting cells. All of that is based on our RNA delivery platform technology, which enables us to selectively direct the RNA expression towards certain organ or cellular moieties. I've demonstrated a little more on the details, on the background in the yesterday presentation, but the bottom line is we can make formulations which lead to highly selective and efficient RNA expression in dendritic cells in the spleen and in lymphoid organs, which is the basis for developing these injectable nanoparticle formulations. We have different approaches to bring this into clinical practice. One is what we call an IMP kit. This consists of This consists of this one, because of RNAs, diluent and liposomes, which are reconstituted or manufactured depending on regulatory constraints right before administration in the pharmacy, and we end up in an injectable RNA nanoparticle drug product. You see that this is a slightly opaque product due to the light scattering from the nanoparticles which have been formed. Furthermore, we manufacture as well what we call ready-to-use products, or the ready-to-use RNA lipoplexes, by using a semi-automated fluid path setup, which is somehow comparable to what has been presented by Precision Nanosystems, who have also posters, a stand, and presentations here. We have our dedicated custom-made setup, which is driven by the special requirements of this kind of formulations. Therefore, we could not use the ones which are available on the market. However, what we can do is we can make ready-to-use product, which is then according to the type of product, either stored in a liquid, frozen, or lyophilized state, according to the requirements for the product. This is an overview over our types of clinical study, or the categories, how we realize our clinical development. We have what we call a fixed WAC approach, where people receive a set of fixed defined tumor antigens which are known. We have what we call a warehouse approach, where we manufacture a warehouse of different tumor antigens. Patients are screened and they receive only the medication with the antigens they are positive for. So they are taken off the shelf after having screened the patients. And we have a strictly personalized approach, which we call IVAC mutanome, where patients are screened. Their antigenic mutations are identified by certain algorithms, and then the personalized medication is manufactured and administered to the patient. Here you see our current clinical study program. We have FixVac, warehouse, and IVAC programs, and we do have RNA to be injected in the locally in the lymph node, and RNA Lipoplex nanoparticle formulations which are injected intravenously in different combinations. And here now I would like to focus further on the LipoMerit study, which was the first one which has been initiated for the intravenously injectable product. Here we have 4 different tumor antigens, which are all prepared as RNA Lipoplex nanoparticles, which are injected subsequently into the patients. This is the study outline. We have 4 different sites in Germany using intra-patient escalation to target doses between 14.4 and 100 micrograms of RNA. We inject the 4 RNA products consecutively, and the aim is to investigate safety and tolerability and to search vaccine-induced antigen-specific immune responses. This is where we stand currently. So as you can see, here are the doses. So we started with very low doses, and now we are already at the level of 100 micrograms of RNA per patient. The expanded cohort is ongoing. And what we can say for the time being is that the 15 patients who have been treated with the dose levels of up to 100 micrograms tolerated well the medication. There were some mild to moderate adverse effects, which more or less are expected because they are in line with the intended immunological response. What we found is, which may be interesting, is that there were large intra-patient variations with respect to the immune response, which may be due to patient-individualized coherencies. This is an example on how we do the immune monitoring, and what you see is that in that case that the response with respect to the displayed antigens here is increased by the medication. And summing up here, you see what we can say so far. We have treated 15 patients for the time being. The expanded cohorts are ongoing. We think that the medication is safe and well tolerated. We have some responses like increase of body temperature, fevers and chills, which is typical for vaccination, but there's no dose-limiting adverse effects. And in the end, we can say that we induce immune response both systemically and specifically towards the antigens we have been vaccinating for. And with that, as my time runs up, I would like to thank the people who participated, and thank you for your for listening to me, and I'm happy to take questions.
Thank you for stopping more than 1 minute ahead of time. Let's go ahead. Question?
The mutational pattern in the tumor is changing very fast within time. How do you meet this criterion? Your mutational analysis must be repeated after which time? How can you tune this with the mutational frequency within the tumor?
Yeah, that's of course a very good question. In fact, we want to find it out. So currently, with this study I presented to you, we have fixed vaccines, or we vaccinate for fixed tumor antigens. For the personalized branch, we could adopt, so patients are sequenced, and then the most antigenic mutations are identified. We could repeat this. But as we have not proceeded so far, we have to find this out within the clinical study, at which stage this could become necessary.
Question here.
What is the lipid composition of your liposomes?
It's lipids that are readily available on the market. It's categorically called Tumor and one called DOPE. This is published in our Nature paper. You can find more details on that.
Right. Do they induce any cell death?
The lipids?
Yes.
Not in our settings, as we use very low doses and overall negatively charged particles. So the general concern on cationic lipids is well known to us, but we did not see any toxicities related to the lipid carrier with our settings.
So your cell death is on what cell you're looking at?
Can you say this again?
When you're looking for your cytotoxicity.
We didn't get any indication for toxic effects of our lipids.
So have you checked that on primary T cells?
I would need to ask our preclinical colleagues. I would think they have looked at it, but I should check with them.
I have a question. So what is the time frame from the time you inject to the time you get the antigen presentation? I mean, of course, the mRNA should be active very quickly to produce the proteins, but can you sort of give some idea?
This should be, actually, I don't know by heart. It would be like something like few weeks when we first have the first monitoring, something between 2 and 4 weeks, but I would cross-check this with our clinical people. So we have fixed time points when we do the immunomonitoring.
Okay. I have another question. So you're going either directly into the lymph nodes or IV. And since the aim is to get into the dendritic cells, can you not do this sub-Q and have sort of the same effect?
We think that going IV is much more useful than going sub-Q or intramuscular or even local, because we have a much higher compartment of target cell which we can address. So in principle, you are right. Vaccines are usually administered sub-Q or intramuscularly or whatsoever, but they have maybe a slightly different mechanism, because cells may take up the peptides and then they may migrate. That's not the case with our medication, as we have a large molecule which rapidly degrades. So we make use of the advantage of being able to address a much higher population of antigen-presenting cells, and in fact the injectable drug product is much more potent than the locally or the intravenously is much more potent than the intranodally injected product.
We have a question here.
Did you look to the T cells in these individuals before and after your application? So you are trying to induce an immune response, but you have the antigen before, so the problem is that the T cells are kind of exhausted. So can you get try to get a recall response before and after and see if they have some differences in the phenotype?
We have a detailed immunomonitoring program where we look at specific activation of T cell for the antigens we vaccinate for.
Yeah, but the phenotype of the cells is different. Do they have different surface molecules, different costimulatory, or secrete different cytokines when you give your vaccine?
Maybe I don't understand exactly the question, so maybe we can follow this up after the session.
Okay, any more? I have one last question then. You've treated 15 patients. Any indication of response in these patients?
Well, I'm not the clinical expert, so I can't answer. What we do see is the indication of the specific immune response. What I understand is that the patient population is not appropriate to get this information you just mentioned. So we have to continue and see. Some people are stable, having stable diseases, but I personally, as I'm not a clinician, cannot judge if this is to be correlated to the drug or not.
Okay. All right, thank you very much. We should move on.